Assalamualaikum warahmatullahi wabarakatuh. My name is Nurish Iman Kamilia binti Muhammad Iswan, metric number 2519772. Together with my group members, we will be presenting our topic entitled The Impact of Complement System on Cancer. But first, let's talk about cancer. Cancer remains one of the leading causes of death worldwide and continues to be a major global health challenge. As shown in this slide, the immune system plays an important role in recognizing and eliminating cancer cells. One of the key components involved in this immune response is the complement system, which part of the innate immune system. Recent study has shown that the complement system has a dual role in cancer. On one hand, it can suppress cancer by helping the immune system destroy tumor cells. It is a good complement. On the other hand, it can also promote cancer growth and contribute to immune evasion, which is bad complement. Therefore, understanding the relationship between complement system and cancer is important for developing more effective therapeutic strategies. To better understand its role in cancer, let us first look what exactly the complement system is. The complement system is part of the innate immune system and consists of more than 30 plasma proteins circulating in blood and tissues. These proteins normally exist in an inactive form called zymogens and become activated through an enzyme cascade, which amplifies the immune response when the body encounters pathogens or abnormal cells. The complement system can be activated through three pathways: the classical pathway, lectin pathway, and alternative pathway. Although these pathways are activated differently, they all lead to complement activation. As shown in the diagram, activation of this pathway lead to C3 activation and followed by C5 activation, and ultimately the formation of membrane attack complex or MAC. The complex system protects the body through three main functions. First, opsonization, which marks target cells for phagocytosis. Second, inflammation, which recruits immune cells to the site of infection or damage. Third, cell lysis through the formation of the membrane attack complex, which can directly destroy target cells. Because of this important function, the complement system plays a significant role in cancer immunity. With that understanding, I will hand over to the next presenter.
So, for the next part, we are going to explain about the good complement system. So, what is the good complement system? The first one is the opsonization. Cancer cells are often sneaky. They look very similar to healthy cells, so your white blood cells might ignore them. So, how does they work? First one, complement protein like C3b act like GPS tracker. They bind to the surface of the tumor cells and marking it. After tag, immune cells can easily spot the cancer. Without this tag, the immune system might just pass the cancer cell by. So for the next one is membrane attack complex, which is MAC. First, the step is a series of proteins assemble themselves on the surface of the cancer cell. They form a ring-like structure called the membrane attack complex, which creates a physical hole in the cancer cell's outer membrane. The content leaks out and water rush in. The process is called complement-dependent cytotoxicity. And for the last one is anaphyltoxins, which is the complement protein breaks down during the battle. They release small fragments, which is C3a and C5a. These fragments act as alarm signals or sentries. They float through the tissue, attracting other immune cells to the scene of the tumor, and then they wake up the dendritic cells, which carry piece of tumor to the lymph nodes to train T cells how to recognize and kill this cancer more effectively.
Assalamualaikum. My name is Iridina Dayana binti Zulkifli with metric number 2519604. I'll present about the bad complement system. There are two main stages of tumor development. First, tumor initiation, which causes DNA damage. Second, tumor promotion, which stimulates growth and proliferation of cancer cells. Tumor initiation. This happens in three main steps. Step 1, anaphylatoxin pool. When complement is activated, it splits into small pieces, which is called anaphylatoxin, specifically C3A and C5A. C3A and C5A attract immune cells like macrophage and neutrophils. Step 2. Oxidative burst. The C3A and C5A signal the immune cells to release reactive oxygen species ROS and reactive nitrogen intermediates RNI. Step 3. DNA damage. The ROS and RNI are highly unstable molecules and can damage the cellular DNA. Second stage, tumor promotion. First, direct mitogenic signal. Cancer cells express receptor for C3A and C5A. Once C3A and C5A bind to the receptor, they act as mitogens. Mitogens mean signal that tell the cell to divide. Next, sublytic MAC. Sublytic MAC stimulates cancer cells to replicate and produce inflammatory proteins rather than dying. Next, angiogenesis. Angiogenesis is the formation of new blood vessels from existing blood vessels. As a tumor grows, it runs out of oxygen. This condition is called hypoxia. Hypoxia triggers cells to lose their complement protecting shields, leading to more complement activation. This complement activation causes angiogenesis to feed the tumor with oxygen and nutrients.
Assalamualaikum warahmatullah. I am Amanda Sofia with metric number 2510308. Now, let's take a look at how cancer cells overcome complement system in human body. Normally, our complement system forms membrane attack complex or MAC to punch holes in target cells to destroy them. But cancer cells have their way to shield themselves, specifically by overexpressing the membrane-bound complement regulatory protein, also known as MCRPS. Here, I list out three main MCRPS that we are gonna go through one by one. The story begins when the cancer cells upregulates the expressions of CD55, which directly increases the density of CD55 molecules coating the cancer cell surface. Now, when the complement system tries to activate on the cell surface, it relies on one enzyme called convertase. CD55 steps in and forces their catalytic subunits to fall apart prematurely. And by doing this, it will speed up the natural decay process of convertase before they can even do any damage. Because those enzymes are broken down so rapidly, CD55 successfully neutralized the convertase. There is no MAC formation because the cancer cells already prevent the punch holes from being built. Next, we have CD46, also known as membrane cofactor protein. Now, the immune system normally pins C3b and C4b proteins onto the cell's walls to build cytotoxic enzymes, specifically the C3 and C5 convertase, which would normally destroy the cells afterward. However, cancer cells can overexpress CD46 so that it can capture the C3b and C4b proteins, acting as cofactor, and then present them to factor 1. Here, it holds the immune proteins in place so that factor 1 can cleave C3b and C4b, rendering them completely inactive. As a result of this cleavage, our complement system fails to build the complement convertase. And without those convertase, the immune response is completely neutralized right at the cell surface, allowing the cancer cell to survive. Finally, we have CD59, also known as protectin. Now, let's say if the immune system successfully bypassed the two previous checkpoints, a C5b8 complex will be generated on the cell membrane. The job of this complex is to recreate multiple copies of C9, which are circulating plasma proteins. Under normal circumstances, these C9 proteins string together, forming a rigid ring, which then establish MAC. However, they overexpress CD59, will intercept the process by binding to the C5b8 complex and blocks the polymerization of C9, and eventually stopping those C9 proteins from locking together to complete the ring. As a result, we see a complete failure of cell bursting. The physical integrity of the tumor cell is maintained, resulting in zero lysis. This leads to the survival of the cancer cell.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. My name is Iftidamia binti Ahmad Shaifulizam, metric number 2515686. Next, I'll be presenting about a recent study that was published in the Journal for Immunotherapy of Cancer, specifically focusing on combining complement blockers with immunotherapy. So, as previously stated by my group mates, the complement system releases small inflammatory fragments called C3A and C5A into the tumor microenvironment. So this is a problem because instead of destroying the cancer cells, these fragments will produce an invisibility barrier around the cells that will block the immune system from working. So as a solution, the scientists from this research discovered new therapies that are responsible for targeting and blocking the C3A receptor and C5A receptor 1. So when these receptors are blocked, then they cannot build the invisibility shield around them. So with the blocking of these receptors, and combining them with standard immunotherapies like anti-PD-1, as you can see in this diagram, then the cells or the immune system will effectively pivot back into the anti-tumoral pathway, where it will allow the infiltration of immune cells, allowing the T cells to identify and target the cancer cells and destroy them. As a whole conclusion, we know that the complement system is a powerful element in our immune system. On one hand, when working correctly, they will use the CDC to destroy cancer cells. On the other hand, when hijacked, the system will release C3A and C5A that will create an invisibility shield around the tumor. So, as a strategy to defeat the immune system, the cancer cells will overexpress regulatory proteins like CD55 and CD59 on their surface. So, this will act as a stop sign and it will shut down the complement attack before it can puncture the cell. Thank you very much. That's all from us.